Hello, everyone. I hope everyone is having a great, wonderful April. My name is Carl Geiser, and I'm a master's student here at Loyola University Chicago. My research focuses on wastewater contaminants and the effect they have on freshwater microbial communities. So one common contaminant that we focus on is microplastics. As you might already know, microplastics are small pieces of plastic debris, approximately 5 millimeters or less in size, and are typically created when the larger pieces of plastic items break down. A common way that these microplastics enter wastewater is from washing synthetic fabrics in like a washing machine. In addition to microplastics, we are also interested in triclosan. Triclosan is an antimicrobial agent that has historically been added to a wide range of consumer products, ranging from personal care products such as soap and toothpaste to more durable goods like kitchenware, pet toys, and even athletic wear. I will mention that there have been additional FDA restrictions on adding triclosan to personal care products such as soap and toothpaste, but currently there's no regulation on how triclosan is used on durable goods such as athletic clothing. So many of these products containing triclosan are used and maintained in sinks and washing machines, which leads to the triclosan going into wastewater. Studies have shown that wastewater in the United States contains a varied but substantial amount of triclosan, with more urban environments typically having a higher concentration, likely due to a greater density of people using these triclosan products. Triclosan is typically added to durable goods because it absorbs to plastic surfaces and provides an antimicrobial benefit, but it also has the potential to encounter microplastics in wastewater and absorb to them there as well. One of the reasons we care about triclosan is that there is a potential link between triclosan and antibiotic resistance genes. Microplastics can also travel very far when they enter wastewater, so you can imagine that triclosan absorbed microplastics could potentially spread microbes that have antibiotic resistance to places where those microbes or genes would not be present. Before we can address that, however, as a first step, we must first understand how the triclosan is affecting these microbial communities that colonize microplastics. And today I'm going to briefly discuss the composition of how these microbes are affected on triclosan absorbed plastics. So briefly, methodology to test how these triclosan absorbed fibers affect these microbial communities that grow in microplastic. We cut up 3 common types of microplastic fibers found in clothing: acrylic, nylon, and polyester, to approximately 5 millimeters in size. We then prepared a triclosan stock solution in isopropyl alcohol and exposed each fiber type to the solution for 24 hours. Later, we tested the triclosan concentration on the fibers to confirm that triclosan was absorbed. We then collected water from the North Branch of the Chicago River at Harms Woods. And finally, we took the triclosan exposed fibers, in addition to control fibers without triclosan, and weighed them out to about 50,000 fibers per sample and inoculated them in small microcosms containing 80 milliliters of water from the Chicago River. The incubation period was 30 days on an incubator shaker with shaking, and they were continually shaken at room temperature. And every 48 hours, the lids were removed for 10 seconds to provide aeration. There are a total of 5 replicates per sample per plastic per condition, for a total of 30 samples. After the incubation period, the DNA was extracted, the 16S RNA genes were amplified, and the samples were sequenced on the Illumina-MiSeq platform. In addition to the microbial communities on the plastic itself, we also sequenced the microbial communities on unincubated fibers as an additional control. And so moving on to the results here. Now I want to show you the results. I first want to discuss the diversity metrics of the data. The first thing I'd like to describe is the graph on the right, which is a PCOA plot comparing the beta diversity between all of the samples that were exposed to triclosan and the control samples that were not.
1 minute.
During my analysis, I found that there's a statistically significant difference between the 2 types of samples. This PCOA plot represents all samples, but this difference is also apparent if I only consider specific plastic types, which I did not show here due to space limitations. This data indicates that the triclosan exposed microbial communities have a different composition from the control microplastics. The alpha diversity information is more nuanced. Here at the bottom, I have 3 separate graphs representing each plastic type. For each graph, I chose to show both Chow-1 richness and Shannon diversity estimates. First box plot on the left represents the estimate of the unincubated plastic, while the 2 box plots on the right represent the incubated plastic. The triclosan exposed plastic is displayed in green, while the non-triclosan samples are displayed in red. The Chow-1 estimate shows that incubation typically increases the richness, but the triclosan exposure appears to lower the Chow-1 richness estimates in the acrylic and nylon samples, but not the polyester. Shannon diversity typically does not change much between the unincubated and incubated samples in acrylic and nylon. Um, this seems to meet my expectation that triclosan is going to reduce the richness and diversity of bacteria. However, polyester bucks the trend. I'm not entirely sure, but I suspect it has to do with surface chemistry, and it would merit further exploration. Just briefly about the differential abundance. There's a lot to process here, and I don't have the time to discuss specific bacteria, but I'm just going to go over the bird's eye view. There are, once again, 3 different graphs of each plastic type, and what they're showing is a difference in the number of recovered 16S RNA gene copy numbers for different organisms expressed as log-2 fold change. The group on the left are less abundant than triclosan exposed fibers compared to the control fibers, while the groups on the right are more abundant. Of note, the cyanobacteria and the proteobacteria all have gene copy numbers between the 3 plastic types, which seems to indicate that the triclosan impacts certain groups more than others. Of course, more exploration will need to be seen if these groups are more likely to contain antibiotic resistance genes, and will be a topic for further study. Thank you for taking the time to watch my lightning talk. I hope you have a wonderful day.